I would like to introduce the first speaker of this session, Dr. Richard White. So he's a professor at the University of Oxford and an adjunct faculty member at Memorial Sloan Kettering Cancer Center in New York. His lab is interested in the intersection between developmental biology and cancer biology. And Rich will talk about oncogenic competence, which is the third required ingredient after the activation of oncogenes and the loss of tumor suppressor genes for normal cell to become cancer. So without any further ado, I set the stage then for Rich. So please, you can start whenever you are ready.
Great, thank you. All right. Hopefully that looks okay.
Perfect.
Right. Great. First, thank you for having me. It's fun to do this type of conference and think about these problems. And as Mohammed said, I'm going to talk about this concept of oncogenic competence today and describe some of our unpublished work in thinking about this problem. So we've now known for a number of years that mutations in oncogenes or tumor suppressors are relatively common, surprisingly common in normal tissues, but not sufficient for tumor initiation. I think one really nice example of this was work from Peter Campbell's group at the Sanger, where they took eyelids, normal eyelids from donated patients, and they did multi-region sequencing on these. And what you can see on the bottom is that mutations in a variety of different genes, encompassing many different transitions and transversions, are actually surprisingly common in this tissue. And now there have been many other studies, recent work from Mike Stratton's group, showing, from Charlie Swanton's group in lung cancer, showing that this seems to occur with a reasonable frequency as well. And so that has interested us for a very long time, actually, in thinking about what's the other ingredients that initiate tumors above and beyond somatic DNA alterations. And my lab has been working on this for a number of years. Oops. And the disease I'll talk about today is in melanoma, which my lab is primarily focused on. So melanomas are skin cancers that arise from pigmented cells called melanocytes, or more commonly from their neural crest progenitors, which are the embryonic progenitors of the melanocytes. Both cells can give rise to melanoma. It's just a question of frequency. From a genetic standpoint, what makes them very interesting is that they're extremely heterogeneous at the single cell level, and this includes at both the genetic as well as the epigenetic level. So there's a lot of substrate there for Darwinian selection to occur. But despite this heterogeneity, what we found a number of years ago is that if we take, in our case, we were using zebrafish for studying this, and we've done this in other systems as well. And I think one of our important findings is that if we take canonical oncogenes like BRAF or knock out P53 or even PTEN, one of the really important findings that we had is that it's really the progenitor cells, neural crest or melanoblasts, shown in the two panels here, that are capable of forming tumors in this case, whereas the melanocytes, driven in this case by a TYRP promoter, can form tumors, but very rarely do so. And when we mechanistically dissected why that was the case, the reason was on the bottom here, see if I can turn my laser pointer on, was that when we profiled the activation of oncogenes or tumor suppressors, what we found is that neural crest cells in particular, which are highly susceptible to tumor formation, have a very robust transcriptional response to the DNA mutations. And similarly, in the melanoblast, we saw a similar thing, whereas the melanocytes, which almost never form tumors, they have essentially no transcriptional response to the somatic DNA alterations. And one of the really surprising things in the study was that this was in spite of the fact that when we activate the oncogene, we have the same level of phospho-ERK signaling, meaning that signaling is completely intact in these three cell types, but they don't have a transcriptional response to that signaling network. And in work I'm not going to talk about today, we eventually worked out the mechanism by which this occurs. which is through a combination of transcription factors and chromatin factors in this one called ATAD2 that endows the cell with transcriptional plasticity. And so when we published this paper a couple of years ago, we used the term oncogenic competence. And essentially that term, as we used it, was meant to encompass all of the factors above and beyond DNA mutations that enable tumor initiation. And that can, encompass both cell intrinsic things, and as I'll talk about today, cell extrinsic things that can push a cell over the edge to becoming a tumor, given the exact same genetic mutations. So why we began thinking about cell extrinsic forces, I think many of us are thinking about this, really had to do with our first transgenic model that we built many years ago in the zebrafish. So this was a model in which we had activated BRAF in the melanoblasts and inactivated P53. And what you can see is that these three genotypes do not form tumors, but these animals readily form tumors. 100% of the animals in the melanoblasts form tumors. But I thought one of the very curious things was that these animals, despite having these DNA mutations everywhere in the body, right, every melanocyte had these same exact genetic mutations, generally only form tumors in one location, right? So we got generally one tumor per animal in highly stereotyped locations. And we always thought this was very curious and suggested to us that there had to be likely local anatomic or microenvironmental influences that were influencing why the tumors arose where they did. And this is a question that we were always very curious about. And so when we began thinking about how to approach this question, so on the top we had shown that the cell state clearly matters for the response to the oncogene. The problem that we were faced with, and I think we're all faced with, is that the components of the microenvironment in each anatomic site is very, very varied. It's very composed of many, many different cell types. I'm showing just a couple of the cell types here that we've looked at in the complex tumor microenvironment. And so I think there was a technical sort of challenge here to figure out how do we begin to analyze these at a functional level to understand these types of interactions and how they relate to oncogenic competence. Because the interaction space is so vast, right? We wanted to first develop a technical tool to get at this question. And so in the melanoma, obviously initiates in the skin, and we knew from lots of work that these early melanomas are primarily surrounded by another cell type, which is the keratinocyte. And the keratinocytes make up the vast majority of our skin surface. Melanocytes are actually relatively rare in the skin. And so it seemed logical to us, and other work had supported this idea, that the keratinocytes in the skin might actually be a dominant factor in determining whether a melanocyte could turn into a cancer or not. So we decided to focus on that cell. But still, there are millions and millions of keratinocytes in the skin. And so to develop the system for essentially tracking these interactions, we were inspired by the biology of melanocytes and keratinocytes in normal biology. And this is a really fascinating biology that has to do with the tanning response. So when we go out into the sunlight, sun hits the keratinocytes, which is the superficial skin. The keratinocyte then sends a signal to the melanocyte to pump out an entire organelle into the to the keratinocyte. And that actually is what's responsible for the tanning response. And this is this really cool movie in which you can see, hopefully the movie plays, where this is an individual melanocyte injects through this dendrite its melanosomes, an entire organelle, into the keratinocyte. And we thought this was fascinating because it's not just melanosomes that get transferred, it's many other extracellular vesicles as well undergo this injection procedure from one cell type to another cell type. And so with this in mind, we developed a system, a fate mapping system for cell-cell communication based on this biology. And this was largely inspired by work from Jakob and Wienen's group, where they had developed this really clever Cre-Lox-based system for doing exactly this. So in their system, you have a donor cell that has a form of Cre recombinase that is fated for extracellular vesicle accumulation. When that cell donates or exports its vesicles, it will have Cre along with many other things in it. And then that will be taken up by a recipient cell that has a FLOX reporter in it. And that reporter can be, in this example, a switch from DS red to green. So when the cell takes up Cre from an adjacent cell, that cell will flip from being red to green. And so we were basically inspired by this type of approach based on what we knew about melanocytes and keratinocytes, and adopted it for our system. So just to walk you through how this works, we developed zebrafish transgenic animals that had multiple genetic mutations in the melanocytes, such as BRAF V600E, P53. We then engineered in this exportable Cre under the melanoblast-specific promoter MITF. So this would be our donor cell. And then we engineered the keratinocytes using the KRT4 promoter to have a FLOX cassette that went from a LAC-Z stop cassette. And if it took up Cre, it would then switch to red. And so the idea would be that any cell that was communicating with these nascent melanoma cells would turn red. And that was sort of a fate mapping system. And this is what the animals look like. So I'm just highlighting one particular area of the animal right here. So in this particular example, we see a single melanocyte, false colored in yellow here. And this is a single keratinocyte that is in direct communication with this transformed melanocyte. So this melanocyte has BRAF V600E, it has P53 loss of function. And now you can see this is the sole keratinocyte that is in direct communication. There are lots of keratinocytes in the rest of the animal, but it's surprisingly few that are in this form of direct cell-cell communication. I think we're underestimating it because these were mosaic transgenics, but I think no matter how many times we did this experiment, it's actually relatively few keratinocytes that seem to undergo this switching phenomenon. So this led us to do, I think, a really important experiment, which was to ask, well, these are rare keratinocytes, are they important? So Mohita, who's the postdoc in the lab leading this, changed the cassette where everything else was exactly the same. They had BRAF, P53, they had the Cre, but now she changed the keratinocyte FLOX cassette that it would switch, in this case, from GFP to RFP, but at the same time would then express a cell ablation cassette. And we used, in this example, diphtheria toxin A. We've also done it with nitroreductase and get the exact same results. The idea being that any keratinocyte that is in direct communication with the melanocyte will ultimately undergo cell death. And then what we read out from the experiment was how many nascent melanoma cells survived if we got rid of that small population of keratinocytes, and that's shown on the right here. And what you can see is that if we do this in the absence of Cre or in the absence of DTA, we get lots of transformed melanocytes that have BRAF and P53. But if we ablate this very tiny number of keratinocytes, now all of those nascent melanoma cells die in turn. And this is not just due to the DTA leakiness, because we did it with other cell ablation cassettes. It's something very specific about being next to this population of keratinocytes that seem to be required for the ability of these initiated melanoma cells to survive. So that gave us a lot of, sort of confidence that these keratinocytes were important somehow, but we wanted to know how they work, right? What's special about those keratinocytes? And the way we went about this was to adopt our zebrafish system to a very similar co-culture system in which we set up the exact same FLOX Cre system in which we had melanoma cells and keratinocytes in co-culture. We could observe switching occurring in the culture, and then we applied a small molecule library, the LOPAC 1280 library, to find molecules that would target pathways that either increase the amount of switching or decrease the amount of switching. And we did the whole screen, and this was the result of the screen. So on the x-axis here are different molecules that came from the library. DMSO is set as a control of 1.0. And what you can see is that a number of molecules in the library markedly increased the rate of communication between the keratinocytes and the nascent melanoma cells. And what was very interesting is that of the top 10 hits from the screen, 5 of them all converged on a single pathway, and that pathway was the GABA-A receptor pathway. And so, in screens, when we see 50% of our top hits all converge on the same pathway, that tends to indicate it's not going to be a false positive, but hopefully is a real hit. And that obviously got us really interested in GABA as a communicating molecule between keratinocytes and initiated melanoma cells. So just to very quickly review GABA, so GABA is typically produced in the central nervous system. Presynaptic neurons express enzymes called GAD1 and GAD2 that produce GABA. That gets packaged into vesicles. GABA is then released into the synaptic space and binds to either GABA-A or GABA-B receptors on the postsynaptic neurons, where they hyperpolarize the cells and lead to a chloride influx. And this is what happens in neurons. And so our hypothesis going in was that what our data would suggest is that the melanocyte was acting like a presynaptic cell and the keratinocyte was acting as a postsynaptic cell. And so this was the idea, and somehow that this was important for tumorigenesis. And so we wanted to test this basic model. And we had sort of reasons to think that this might actually be the case, because in that movie I showed earlier, it's been known for a very long time. If you just look at the morphology of melanocytes or transformed melanoma cells, they are extremely dendritic, right? So they're not quite neurons, but they're quite neuronal-like in a lot of ways. So I don't think it was a conceptual leap to assume that perhaps melanoma cells might retain some of this neuronal function, perhaps due to their origins from the neural crest. And so the key experiment, and we did a series of key experiments that I think tried to address this point, was if this type of communication indeed was important for melanoma initiation, we created transgenic animals in which the keratinocytes, using the keratin4 promoter, expressed a dominant negative GABA-A receptor. So this is a gene called GABAergic2. And what you can see here is, so we expressed it only in the keratinocytes, and then we measured the number of initiated melanoma cells in the zebrafish. And what you can see is that control animals, as we saw before, have many initiated melanoma cells, but that if we express this dominant negative GABA receptor, now we get a significant decrease of this. Not quite as efficient as just ablating the keratinocytes, which is not surprising because there's probably multiple mechanisms involved, but clearly a very significant effect. So we wanted to ask whether this type of GABAergic signaling was present in human melanoma as well, to ensure this was not something quite specific to fish skin. So the way we approached this was to stain a series of human primary melanomas that we had from a tissue microarray, as well as a matched pair of normal skin samples, either from the same patient or from a different patient. And then what we did is we stained the normal and primary melanomas for KRT17, which marks the keratinocytes, S100, which marks the melanocytes, and then the protein gefrin, which marks the GABA-A clustering protein. So GABA-A receptors tend to cluster on the membrane, and that can be marked by gefrin. And what you can see in the top here is that normal skin expresses KRT17, obviously no S100 because there are no melanoma cells, and really no gefrin, right? So they don't really express this. But in the primary tumors, obviously the keratinocytes are somewhat disordered. We have lots of early melanoma cells shown in yellow here. And now we have lots of these keratinocytes that express the gefrin receptor only at the site of tumor initiation. And you can see this on the merged image here, suggesting that there's a bidirectional feedback between the initiation of a true bona fide melanoma in patients and the induction of this GABA-A receptor in the surrounding keratinocytes. And it's been known for a while that these keratinocytes can actually expand upon tumor initiation. So that led us to conclude that this pathway is conserved in human melanoma as well. So a question that we wanted to address was if the hypothesis here is that these are forming synaptic-like structures, synapses are typically clustered along the cell membrane. They shouldn't be present everywhere along the cell membrane. So here what we used was high-resolution imaging in which we have melanocytes marked in gray here, keratinocytes marked in blue, and then gefrin, the GABA-A receptor, marked in yellow. And you can see very nicely is that the vast majority of the keratinocyte cell membrane actually does not have the GABA receptor, and we only see very focal staining of GABA-A receptors in this focal area, very reminiscent of a synapse. And we're not claiming these are exactly like synapses, but they have synapse-like features in this clustering behavior. And if we look more closely at this, you can actually see, so these are melanocytes in yellow and the keratinocytes in purple. And if you zoom in on this, you can see that although melanocytes are very dendritic, their point of contact is extremely small. It's very, very focal where these two cells communicate with each other. So finally, we wanted to ask whether or not this was seen in other ways, like electron microscopy, and we could see very similar things here using EM. So the last thing that I think is important is whether or not these show evidence of electrical coupling. And so to do this, we used a multi-electrode array in which we could actually measure electrical spiking between the regular keratinocytes and the switch keratinocytes and the melanoma cells. And each dot here basically represents an electrical spike. And because GABA is an inhibitory neurotransmitter, our expectation from this experiment was that this would actually decrease electrical coupling. And I thought it would be easiest to just show the movie. So on the top here, these are keratinocytes showing spiking when present with melanoma cells. Each thing is a spike. And then you can see in the presence of these switch keratinocytes, which have high GABA signaling, now we see a very clear decrease in electrical coupling between the two cells. So this seemed to confirm that GABA indeed was active between these two cells. And so the question really then became, is GABA then sufficient to endow oncogenic competence? And so to do this experiment, we took our original fish, in which we induced melanomas in a subset of the fish, but then we created animals in which we either overexpressed the GABA synthesizing enzyme GAD1 or GAD2. We also did knockdown experiments. And the readout from this experiment was, first, how well does an animal form a tumor at all? And it's pretty clear that if you overexpress GAD1 or GAD2, many more animals form tumors. But I actually think it's the graph on the right that was the most interesting in a lot of ways. Whereas earlier I had said that basically every time we did this experiment in the fish, we ultimately got one tumor in the fish. But if we now endow the melanocytes with enhanced GABA signaling and read out the number of tumors per fish, we go from an average of 1 now to an average of 4. And so I think that one of the lessons from this for us was that the genetic lesions in the tumor can sort of predispose those cells, but that a factor from the microenvironment, in this case GABA, can actually now give multifocal tumors and make many, many more of these cells susceptible to tumorigenesis. A very clear example of oncogenic competence induced not by genetic changes, but by something from the microenvironment. I think for the sake of time, I'm going to skip over the details of how we think the keratinocytes are doing this. I'll just summarize by saying that what we found is that these switch keratinocytes that are GABAergic secrete a number of growth factors that then feed back on the melanoma cells to promote their proliferation. In particular, LIF seems to be a very critical factor for what the keratinocytes secrete into the extracellular environment, which then promotes the proliferation of these nascent melanoma cells. And so I'll just summarize by saying, I think what we found in our earlier work, we showed that the developmental state is a critical determinant of how well a cell can respond to oncogenes and be competent to form a tumor. And I think what this new work shows is that above and beyond that, a very select population in the microenvironment, the keratinocytes, can respond to GABAergic signaling. This hyperpolarizes those keratinocytes. That relates to the secretion of LIF, which then sort of pushes that melanoma cell over the boundary to become a tumor. And we think there are many other factors here, obviously besides GABA and LIF, that are probably coming from the microenvironment. The other thing I think we established is this really nice fate mapping system, and Ilaria has had others to look at this type of cell-cell communication. And I think that the more tools we have to look at cell-cell communication, the more mechanisms like this I think we'll uncover. And so, I think competence, it has many factors, developmental state, anatomic position, where the cell's located, and we published about this as well, and obviously the microenvironment, and we're excited to learn more of the factors from the microenvironment that can do this. And I just want to take a minute to thank Mohita Tagore, who's a postdoc in my lab, who did all of this work, and really the beautiful genetics and imaging that she did. And I'll take questions, so thanks very much.
Thank you very much, Rich, for this clear presentation. I see that we have a couple of questions in Q&A and question from a live question. Then, Thomas, maybe you can start with the live question.
Thanks, Rich, for a great talk. Very interesting. I had a question about the location on the body where these tumors arise. I guess it's a two-part question. Is there a stereotypic location where these melanomas typically arise? And is there anything special perhaps about the keratinocytes there?
Yeah.
And actually, I have a follow-up question to that, which is related to sun exposure. So if you do something, UV exposure, if you do something that promotes the melanosome deposition, do you actually promote this GABAergic interaction as well?
Yeah, this is both great questions, and they're kind of interrelated in a way. So let me address the first one really quick. We think that we've done experiments where we did single cell sequencing of skin from anterior locations where the tumors typically arise versus, let's say, the fin, which is obviously a completely different microenvironment. They're wildly different from each other, actually. So, and we're currently delving into exactly why they're different. But that actually relates to the second question, which was that in evolution, if you think about it, the dorsal keratinocytes on the fish, or what would be like our face or back in a human, are exposed to far more sunlight than things on the palm of the hands or the fins of the fish. So we've actually recently set up a light box experiment where we're exposing them not to UV light, but actually to visible light, which is the more common thing. So those experiments are ongoing. We have a light box set up and we're testing it right now.
Great, thanks.
Thank you very much, Rich. I have also a question myself.
Sure.
Is the process that you described is important for the nascent melanoma? Do you think that similar process may also be found in the metastatic side? Like some elements of the niche that may help, the melanoma cells grow in a distant organ.
Yeah, this is a really good question. So at least for GABA, what we found is, those animals that had these multifocal tumors, but surprisingly, they didn't have increased metastases, actually. So that leads us to conclude that at least for GABA, it seems to be quite specific to that niche and perhaps not other niches. But the one place that we didn't look is the brain, because the fish melanomas do not metastasize to the brain. And so we actually weren't able to look at that. But that would be the other logical place where this could play out. But we just couldn't look because we didn't have the tools for it.
Thank you very much. We have two questions on the chat. So this is from Julie Pankan. The question says, it seems that GABA activation not only increases the number of tumors, but also changes their localization. Any explanation?
Yeah, we're trying to figure that out. I don't have a great answer. We noticed that also, actually. One of the most striking things, and I don't know if you're a fish person or whatever, but tumors in fish almost never develop on the ventral part of the fish, actually. But yet, when you overexpress GABA, we get ventral tumors, which is exceptionally rare. So I think that it must have something to do with some bidirectional crosstalk. I don't have an answer for this, but it was one of the most striking things we observed as well.
Thank you. So we have then second question is from Waihana Iriondo Martinez de Zuazo. So it says, hi, Richard, great talk. Did you find that areas where the tumors formed in one tumor fish had more baseline GABA signaling?
Oh, that's a good question. We did not look at that actually. We have single cell data, so actually we should just look at that. That's a fascinating question. We didn't think about, is there just differences in GABA inherently? If you email me, I'll give you the answer to the question because we have the data, we just haven't looked. So.
So maybe I ask another question more general about the oncogenic competence.
Yeah.
So is there any example in your field in which, for example, melanocyte may acquire genomic alteration, be it genetic or epigenetic, that may cause the cells to de-differentiate to an earlier stage in the hierarchy, and then they acquire the tumorigenic potential?
Yeah,
in
that's
mammary glands, we have examples. I'm asking about melanoma.
Yeah, so I think the best example is in that first paper I talked about where we described competence. We had looked at this gene ATID2, which is a chromatin factor, and found that if you take a melanocyte and expose it to ATID2, it de-differentiates and becomes unpigmented. So I think that that's a very potent example where a single chromatin factor can actually push the cell more towards being neural crest-like and way more susceptible to oncogenesis. We haven't looked at other chromatin factors, but that's the best example.
Yeah, thank you very much. So I see that there was a hand somewhere. Dana, please go ahead.
Thanks. So just to follow up on that, LIF would actually induce also a sort of the differentiation in the melanocytes. Do you think they become then more neural crest because of reprogramming or whatever?
Yeah, that's one of the ways that we think it works exactly, is that it probably pushes the cell towards a slightly less differentiated state. You know, in our fish system, we were starting with a melanoblast promoter, MITF, so the cell is already primed in a way. One experiment we've talked about, but we've not done, would be to take a fully mature melanocyte, overexpress GABA, which then would activate this LIF signaling, and see if then that cell is now competent to form a tumor based on the mechanism you said, is that it pushes the cell to become less differentiated. I think that would be a fantastic experiment.
Without any additional oncogenes, you mean?
Exactly, right? So take a melanocyte, give it GABA, and then it'll get lift from the keratinocyte and see if now it can form a tumor. Yeah.
Thank you. I see that there is a question from Dirana. Please go ahead.
Hi, thanks. Great work, Richard, as always. I was curious about the status of this signaling program between the keratinocytes, LIF, and GABA in nevi, in benign lesions versus malignant, and whether you think some of this biology may actually dictate whether there's this stop in the tumor process, call it senescent or whatever, lack of epigenetic competence to become malignant, and whether you found differences in these two settings of premalignant and malignant biology.
Yeah, the best data we have is when we looked in at least normal skin, which don't really have nevi, we didn't see much evidence of GABA signaling. Getting samples of nevi to do the exact thing you suggested, we tried and failed actually to get good human samples to really analyze this in detail. One way that we could experimentally do it would be to take away the p53 mutation. So do BRAF, wild type p53, and then ask if GABA actually plays a role in pushing that cell either to becoming a tumor or preventing that from happening. We haven't done the experiment actually, and we have not been able to stain the human samples properly.
Thank you.
Yeah.